So we'll get started with tonight's topic is going to be vegetative propagation. Most of our presentations are kind of going at the higher level, just giving overviews of different topics. Because there's so many topics or things within our topics that we can't really go in too much detail because we'll take too long. But this is kind of like an introductory series, and then we'll probably potentially add on more in-depth programs later on. And so vegetative propagation, another word for it is asexual reproduction, or we're producing new plants that are genetically identical to the mother plant or that plant that we're trying to take that plant material from. This is possible because of meristematic tissue. I'll go into what a meristem is a little bit later on. And then two fundamental characteristics make this possible. So totipotency, since each living plant cells has the genetic information necessary for generating all plants or plant parts. And then this dedifferentiation ability, so they're capable of developing these developing cells to return to a meristematic state and then develop a new growing point from that particular spot. And that allows us to generate different plant parts on places of a plant that it never really should have been. And so some of the benefits to using vegetative propagation, it can be easy and convenient, especially if the seeds are no longer available. There's uniformity of clones. If you're taking similar plant material, you're potentially going to have the same stage of growth for your plants once they're ready to go in the ground. And then some plants are seedless, especially stuff like hybrids where they need to be crossed together to get that specific variety. And there's no other means of propagation, especially if you don't have access to the parent material to make the crosses. You're going to have to take a vegetative portion of that plant and try to regenerate it to get more of that particular plant material. And then the ability to combine different species on the same plant is part of the vegetative propagation methods. And there's potential for shorter time periods from when you plant that plant to maturity, and when, for example, fruit trees, when you can get fruit from that tree. A lot of vegetative practices allow for um, speeding up that planting to harvest period versus from seed. And so a few methods that I'm going to go over tonight is grafting, budding, cutting propagation, air layering, doing separation, division, a little bit in tissue culture, and this idea of macropropagation. And so because we were talking about seed production, I want to kind of start with vegetable seedling grafting. And so some of the advantages of vegetable seedling grafting, one of the things is you can avoid or you can provide more tolerant plant material to soil-borne diseases and other things like nematodes if you can graft a good variety onto a more resistant rootstock. There's potential for increased yields, potential for increased plant vigor. Some of the disadvantages include increased costs. You may need additional structures to provide the growing area for these grafted seedlings, specialized labor to be able to make these grafts, and then the infrastructure. And so one of the strategies is the reason why I have this photo of this giant pumpkin is the ability to add additional rootstocks to a growing plant. So you could have multiple rootstock on a single seedling to provide more nutrition to support the vegetative or the fruiting growth of, for example, a pumpkin. And so the different types of grafts that you can do for seedling grafting includes splice grafting, side grafting, a tongue approach graft, and then a hole insertion graft. So this image to the right shows a type of graft that you can do on a tomato plant. And so a splice graft, some of the advantages to splice grafting, it's simple. You don't need to trim a lot of material. And so this bottom half of the plant in that 1C image, you can see that's the rootstock. And so the rootstock, you're basically cutting it at the tip, and then you're splicing on that seedling from the variety that you want. And then on that image in 1D, it's called a grafting clip, and all you're doing is putting on your top and the bottom together and putting the clip to hold it together to fuse that vascular system together and let it heal up, and then you'll get that connection or that graft. The disadvantages to almost all of these you're going to require environmental control where you're minimizing the light, maximizing the humidity, and managing the temperature during that grafting fusion period. A side graft is typically done below the cotyledon versus the splice graft is done above the cotyledon. The cotyledon is that non-true leaf, which is shown holding on that fingernail side on the 1C. Disadvantage is pretty simple. You're cutting into part of that rootstock and you're just putting in that scion wood or that top part that you want on the bottom. And then once it's fused together, you cut off the top of that rootstock. And same thing, requires the same environmental control. And then later on, you're going to have to remember which one to remove when you're removing the tops of the rootstock. And then a tongue approach, the advantage is also relatively simple. You require limited environmental controls. And then some of the challenges include removal of the top of the rootstock 9 days later, and then you have to remove that rootstock of the scion about 11 days later. And so one of the challenges to this one is that you have to be able to put your seedlings in the tray together while they're fusing together because they're living within that cell during that fusion process versus the other grafts, you can basically graft all of your scion onto a whole tray of rootstock and not have to worry about making sure they're planted next to each other during that grafting period. And then the hole insertion technique is you're basically cutting off the top of the scion down to the cotyledon. You potentially use a toothpick, you poke a hole in the top, and then you insert the scion from that graft that you want, or the plant that you want. No trimming is one of the advantages, a little bit more skill to be able to poke it in shallow enough to get a good connection. And it also requires the same environmental control. And so one of the things that you typically need to build when you're doing seedling grafting is this healing chamber. So it could be as simple as this PVC structure with polyethylene, which is plastic that you cover over to create the humidity. And then you can control the light using either weave fabric or shade cloth or some other mechanism to prevent that direct sunlight onto the plant, which will then heat up that healing chamber. And then when you're making the grafts, you want to make sure that those seedlings before making the grafts are hardened off. And so you want to expose them from that very optimal nursery condition out into a little bit harsher condition that includes full sun and maybe reducing the amount of water that you're providing to the plant. When you're making the grafts, you don't want to do it in the middle of the day. You want to kind of do it early in the morning or typically in the afternoon when it's cooler, so it's less heat stress on that plant while that first initial connection is being made. And then maximize the area of your cut surface so you get good vascular connection. And then maintaining the humidity within that chamber from 77% to higher, and then not letting that temperature drop below 70 or exceeding 80 degrees Fahrenheit. And then minimizing that direct sunlight for 2 to 3 days till you get good trigger pressure and connection between those vascular tissue. And then usually you don't put in overhead irrigation because the water that drops onto the tissue could potentially create rotting and diseases, which will then inhibit that connection of that vascular tissue. And so typically the seedling trays will be provided subsurface irrigation, and which could be just a sheet of plastic that the trays are sitting on, and you're just providing a small level of water at the bottom so it can wick it up into the tray to provide moisture to the root systems. And then this is kind of a timeline for seedling grafting. And so you plant your rootstock seeds are planted in 2 to 5 days. Your scions are planted. The seeds for your scions are planted, and then both your rootstock and scions will emerge. And then 10 to 14 days later, depending on the crop that you're growing, you make your grafts and then move them to your healing chamber, where you exclude light. And then as that week progresses, you can increase that light. And then you slowly decrease that humidity within that chamber, meaning you just reduce the amount of water that you're allowing to set in that chamber. And then after that 7 days, hopefully the grafts have taken and you can move them out of the chamber into a regular nursery situation. And then once that has hardened off and you know that the connection has been made correctly, you can take it out to the field and plant it out and it's ready to go. And so this word hardening off, all we're doing is basically prior to your transplanting, you're exposing them, like I said, to increase sunlight and decrease humidity for a few days just to strengthen its tissue. So when you put it out into the field, it's not shocked from going from that high humidity, low temperature situation out into a field situation that typically has lower humidity and higher temperatures. No, like I said, we're just increasing the exposure to light, decreasing the water, and then acclimating it to the elements that it's going to be experiencing out in the field. But you can still maintain a little bit of that support by putting in overhead irrigation and stuff like that to keep it from going into complete shock. And then grafting and budding, mostly for tree crops and woody crops. So primary purpose for grafting is you can potentially get a dwarfing effect by starting with a shorter rootstock and allowing for that mature growth to start at a very short level. And then you can typically increase resistance to disease and insects if you know of varieties of rootstock that has that genetic potential. There's potential for increased structural strength. So you can use grafting on the tree to make the tree stronger by connecting plant material from branch to branch. That's a potential for grafting. Changing the form of a plant. And then basically the main purpose of grafting can be to speed up the time from making the graft to harvesting versus planting a seed. And so many tropical fruit trees, including stuff like mango, avocado, can be used, grafting can be used to speed up that process. Grafting also allows for maintaining the genetics of that scion wood. And so the scion wood is that top part again. And if you were to plant a seed, for example, from a Burposa tree, that seed is not typically true to type. And so if you planted that seed will probably not taste anything like a Raposa. And so if you want that Raposa, you're going to have to either air layer it or graft it onto a rootstock to maintain that genetic variety. And so I talked about this vascular tissue. So this is a cross section of the vascular tissue. You have your xylem, which is that center portion that provides water from the roots to the tips of the plant. And then your phloem, which is that outer area, will provide nutrients from the shoots down to the roots. And so when you're making your graft, you want to make sure you line up this cambial zone with the other from the rootstock to the cambial zone of the scion wood. And that's what's going to connect to make sure that you have that vascular connection going up and down. So grafting, basically the main components of the graft is your scion. This is the desired plant material that you want to have the fruit generated on. And then your rootstock is the stock on which that scion wood will be grafted onto, and that can typically be any seedling or seed that you have available can be planted to use as a rootstock. Another option for rootstock is to take cuttings from a plant and make the rootstock from the cuttings, and then once that cutting is established, you can graft your scion wood onto that cutting. This is potentially important if you know this particular variety is, say, resistant to Phytophthora in the soil, and it's not going to be true to type. So you can take cuttings from that particular plant, make a rootstock out of those cuttings, and then graft your scion wood onto that rootstock and benefit from that Phytophthora resistance in the rootstock while still eating the variety that you've selected is best for your operation. And then this word union or that scion rootstock relationship is very important. We talked about that cambial contact. When you're making the cuts on these grafts, you want to make sure that they are as smooth as possible. So you want to use the sharpest knife possible. And then you want to make sure that you line up those cambial zones as best as you can and make a tight connection with the grafting tape. And then you want to make sure that you have compatibility with your scion and rootstock. And so in most cases, the plants that you're grafting have to be within that same family or genus species. And so citrus to citrus, avocado to avocado, mango to mango, and so forth. And so you won't be able to graft a mango onto an avocado because they're not compatible. And so um, that's some of the limiting factors for grafting. And then continuing on with this meristematic tissue, this is where those calluses will form and make that union or that connection. These cells will connect to each other and kind of fuse together. And then they'll regenerate this connection between that xylem, which is that water movement, and the phloem, which is that nutrient movement. And so scion wood, you're typically looking for plant material um, that is well hardened off. So meaning they're mature wood. Mature meaning that they have mature buds. If something has flushed out new growth already, it's probably too late to take that material for the scion wood. You'll have to wait to that particular leaves harden off and then the buds on that particular shoot harden off and mature. And then when you're taking that scion wood, you're looking for material that is pretty close in diameter to your rootstock. And that could be anywhere from a quarter inch to 3/8 inch in diameter. And so it can be challenging to time the seedling production with your scion wood collection. So you want to make sure that you're keeping an eye on your scions and your seedlings to make sure that you're at the right time to collect the scion wood for grafting onto your rootstock. If they're a little bit off in size, it's okay. I've heard that it's better to have larger scion than the rootstock, but every grafter really has their preference on the size of wood that you're taking um, to to graft onto your rootstocks. And in most cases, the scion wood, as long as you have a bud, you can graft that onto your rootstock. And some people like multiple buds and some like to have just one bud. And so on that scion wood that you have, like for example, these sticks in the photo might have anywhere from 4 to 5 buds. And so this could potentially be uh, a couple scion woods from each one of these sticks. And then your rootstock, it could be roots, branches, or if you have a variety that was from seed and you don't like it, you can actually cut that trunk and then place in the scion in the bark of that trunk, and we call it top working. And different ways to produce rootstocks. And like I mentioned, the cutting propagation can be a way to start rootstocks, and you don't have to use it just seed if that option is available. When you're making grafts, there's a few general materials that you need. So the plant material, which we talked about, was the scion and the rootstock. Then you need a really sharp knife. You can use any knife you have, but it's recommended to use a specific grafting knife. Typically, grafting knives are beveled on one edge, and so the flat edge is the edge that cuts the surface on the scion wood and the rootstock, and the bevel edge should be on the outside facing to remove the wood. That flat edge is going to create a flatter cut on your material. If you use a regular like kitchen knife or a pocket knife, it's typically beveled on both edges, and so that bevel can create gouges when you're making the cut, and that gouge is a little bit harder to connect directly flat onto the surface of each side. And so it's usually not recommended to use a double beveled knife versus a grafting knife, which has that single bevel. And then grafting tape. The old school way that I learned was using rubber bands, but now you can buy parafilm, grafting tape, and other materials just to tighten that connection between the scion and the rootstock. And then there's waxes out there. Um, a few other grafters I know use just plastic bags that they drape over the top of the graft that drapes down past the union. And there's always to keep it from drying out. So these are some of the main materials that you're using to make the graft. Um, Types of grafts, splice grafts, cleft and wedge grafts, whip and tongue, side veneer, bark graft, T-bud grafts, and chip grafts are the common graft types. So a splice graft is probably one of the easier ones to make. All you're doing is cutting a slight angle on your scion and your rootstock, and then you want to make that cut pretty similar on both sides, and that's where you're going to make that union. Once you've connected it, you'll take your rubber band, your grafting tape, and you'll wrap it around as tight as possible to create that connection for the next month or so. And that's going to create the callousing, that cell division and mutation or meristematic tissue to generate that connection. And then hopefully in a month or two, you have that connection and you should see sprouts coming out of that scion wood. When the sprouts come out, it's typically an indicator that things are going well. And then uh, typically after that, a month after that, you can start removing the the wrap, and hopefully you have a good scion connection to that rootstock. A cleft or wedge graft can be used when you're doing top working. So like if we're talking about that trunk area, if it's not significantly huge, you can create this wedge inside that trunk. And you can take your scions, you can put one on each edge, and then you're kind of splitting like that. In this case, because you have so much space, you're going to be using the grafting wax to fill in that area. And then you're going to wrap around that connection to make it as tight as possible. And then that edge that has the meristematic tissue and that cambial zone should fuse to that individual scion. And then you can either keep both of them if they both take, or select the one that looks the strongest, and that can become your main trunk from that particular point. The whip and tongue graft, basically you're making a splice graft, and then from that splice cut, you're going to actually cut down that to make a tongue. And the tongue on both sides are going to interlock with each other, making this connection you can see here. And then you'll go back in with your grafting tape and you'll wrap that connection and do the same thing as a splice graft. Side veneer grafts, you're taking a chunk off the side of that rootstock, and typically you can leave the top part of that rootstock on until that graft is made. Many folks will remove it just to remove the apical dominance off of that rootstock and provide more of that directed energy towards the graft. And so you take out a chunk, you cut your splice cut on top of your rootstock or your scion wood, sorry, and then you would stick it into that side cut that you've made, and then you'll wrap it with your grafting tape and then allow it to fuse together. And then bark grafting is another way to top work trees. And so once you've cut the top of the tree that you don't like, or a branch of the tree that you don't like, you're taking your knife and you're cutting a section off of that bark. Hopefully the bark is not too woody. If it's too woody, it's a little bit harder to pull the bark back. And then you're taking your scion wood and then you're slipping it into the bark. And so when you make your cuts on the bark, you want it the width of the scion wood. And then once you slip them all in, you can take your grafting tape and wrap it away all the way around and tighten it up. And in some cases, if it's really big, you can put in more grafting wax to fill in the spaces so you don't get pockets of air where water can generate. That water is then going to create diseases and potential rot, and you're going to get poor success on your grafting. T-bud is another common grafting technique. And so what this does is on the rootstock, you're going to make a T cut on the bark between the nodes. You're going to cut the top, you're going to slice it down the length of the branch. And then this is a bud from the scion plant or the mother plant that you're taking your wood from. And so that on that branch, every bud, you can cut off a section of it. And all you're doing is removing the bark of that scion wood piece. And you're taking that bud and you're slipping it into this tea that you've made, leaving the bud of that scion exposed like this. And then you would wrap around this part, leaving that bud exposed. And then that bud will become connected to that branch and become a new shoot. And then everything above that, you want to make sure that you remove. And then a chip bud is basically you're just cutting instead of making a T, you're cutting a section of that rootstock, and you're cutting the similar section of the scion wood, and you're taking that from there and placing it onto the rootstock in that section that you cut out. And hopefully they're about the same size. And then that's going to allow for this connection, and you'll take your grafting tape here and wrap it underneath that bud and above the bud to create a good connection. And so this is actually in my backyard. I planted a citrus tree. It was a calamansi, and I didn't like it. And so what I did was I took every branch, or I took about 5 branches on this particular tree, and I did that um, top working on it. And so what I did was I just cut out a piece of that bark. I slipped my scion wood into that section. I use flagging tape instead of grafting tape because I like the stretch of that flagging tape versus the grafting tape. I wrap it around as tight as possible to create a good contact surface. And then after that, I take the parafilm, I wrap it all the way around just to create a good humid environment for the scion wood so it doesn't dry out before the graft union takes. And then usually I wrap it all the way down past the graft union and the flagging tape. And so I removed all the plants, and you can see all the random grafts I made on this particular tree. So this one now has a Tahitian lime. I left one of the branches for the calamansi. I put on a blood orange on this branch. And these branches have tangerines, and then a navel orange on this branch. Um, and then a month later, you can see these shoots coming out of that parafilm. It'll bust right through the parafilm, so you don't have to remove any of that parafilm. Once the shoots have taken, I know that the graft union has pretty much set. And then once I know it's set, I can take off that flagging tape and peel back some of the parafilm. And then you can see some of this white callousing. This is what I mean by callousing, is this white portion. It's like a scab that, like when you get a cut, your scab grows. And then underneath the scab is where the healing is taking place. And so this is kind of what's happening on that branch. And once this graft takes, this eventually becomes the branch. And then what I do from here on is make sure that any buds that come up on this branch below this graft union I remove so that I have more energy going to that particular plant or per variety. So some things to remember for uh, grafting, ask yourself, is that scion and that rootstock compatible with each other? Do I have a good sharp knife that I can make the cut with? Are my scions the right stage of growth? Do I have good mature buds that once I connect it to the rootstock, is it going to be able to shoot quickly right out? If not, it's going to sit there and not be able to generate its energy. And then what method of grafting do I prefer? And so before uh, doing grafts, I would say to just go get sticks and practice grafting the stick back to the stick. So cut the stick in half, make a cut, graft it, and try and pull the two apart. And if you've made it tight enough and the graft is cut well, when you tug on it, it should kind of stay together. Um, and if it does stay together, you know that your union has more chance to be successful. And I would say practice doing that as much as you can before doing it to live seedlings, and that will help to increase your success and not waste seedling material if possible. Next, we'll get into cutting propagation. And so you can do direct field cutting propagations versus generating cuttings in a greenhouse. Uh, One of the crops that or the growers that I've worked with are basil growers, and a lot of times basil is grown directly from cuttings in the field. Sweet potato is another one that's grown by cuttings in the field. You're directly planting the cutting into the field and allowing it to establish a new plant from that particular cutting. And then in the greenhouse, more of those woodier cuttings are typically where you would use the greenhouse situation on a mist bench to make those cuttings. Some of the advantages to cutting propagation, um, yep, older planting material that doesn't produce seeds, you can generate plant material or planting material during this method. And just like airlay or grafting, you can get this earlier harvest date because you're basically rooting mature wood or mature plant material that has the capability of fruiting immediately. And then some of the disadvantages, we're looking at potential disease transfer. And so if your plants that you're taking the cutting material from have a disease, especially viruses, you're taking that virus with you to the next field. And so you want to make sure that when you're taking your cutting materials, you're taking it from healthy plants and you're not spreading diseases to your next field because that's going to set you behind. And then some types of plants or crops have very limited success. And so it may be a challenge to get a lot of plant material during doing this method if you are not familiar with the procedures of that particular crop. And there's possibility for less vigor because seedlings typically have more vigor than these vegetatively produced materials. It can have less productivity going in the field than if you were to plant the seed version of that particular crop. So cuttings is a vegetative plant part that can regenerate the missing part, typically the roots, to form that complete plant. And what it's doing now is forming adventitious roots. So from that cut end in that cambial zone, you have that meristematic tissue that's able to generate or dedifferentiate into root cells and become roots. And then typically, when you're making cuttings, you can use roots, you can use leaves, you can use woody or herbaceous stem cuttings to make these cuttings. So root cuttings, I think one of the more common plants might be ulu. So when you cut the roots in the field, they tend to generate shoots from those roots. And then you can then dig out those roots that have those new shoots on them. I think in some cases you can take the roots out of the field, bringing to a nursery, and you can get shoots developing on those roots as well. And that's what I would consider a root cutting. Leaf cuttings is not very common. Um, in some cases, plants don't produce new shoots from that root end. And so you might just have a leaf, a rooted leaf instead of an actual plant. But in some cases, you'll see these shoots start to generate from that basal end. And then from there, you can start excising plant material off of that leaf cutting. And then your common woody stem cuttings, um, you have these are just ornamental plants. So crotons, panax are some common ones that you can make woody cuttings out of. Panax in most cases, most panax that I work with, we just stick a stick in the field and that would be a direct field cutting. The crotons would be probably more of a nursery setting. And then your herbaceous stem cuttings, which would be like the basil. Some people have done uh, indeterminate tomatoes can be made into stem cuttings. And then when you're making these cuttings, you want to make sure you understand polarity and then making sure that the bottom end of that stem is going into your rooting media and not the top end. And so when you invert it, you're potentially getting shoots growing from the bottom and roots growing from the top. And so you want to make sure when you're sticking your cuttings, you understand which is the top and which is the bottom. When you're selecting your rooting media, there's a bunch of different medias out there. A lot of it is preference and availability. Uh, when I learned to make cuttings, we use mostly vermiculite or a combination of vermiculite and perlite. Now that I do a lot more cutting propagation, I've included stuff like peat and coir. So when we make stuff like ulu cuttings, we're using more coir-based media, and we'll put in a little bit of perlite if we need to. But for all of them, you want to make sure that you have a well aerated, um, good drainage, holds moisture because you don't have roots on it. You want that moisture to keep it from drying out. Um, Typically, you use fresh media every time because you're potentially going to be bringing in diseases if you reuse media. And in most cases, soil is a poor media for rooting most cuttings. And so you don't want to use actual soil from the field. You want to use some of these more lighter, uh, inert media to do your rooting. And then when you're doing a lot of these cuttings, in most cases, you probably want to use a rooting hormone. There's three main types of auxins or rooting hormones out there. Indoacetic acid is the first one. It's not really too common, at least from an availability standpoint. Most growers I work with typically go with something like Hormex. This is indobutyric acid. Hormex makes a bunch of different concentrations. So this one in the image is 16, which is 1.6% IBA. I think it goes down to 0.03% IBA. For the most part, I just use the stronger one on everything because it's the strongest one and it seems to work pretty well on everything. And then some plants actually prefer other types of hormone. And so naphthalene acetic acid or NAA is the other um, form of rooting hormone. And something like Dip and Grow has both indobutyric acid and naphthalene acetic acid. And so you get the benefit of both. And some cuttings just like to have a little bit of that, both of these hormones to stimulate rooting. But it's a lot of trial and error with the plant material that you have to try and um, get them to root. Another important component to rooting cuttings is a mist bench. You can try and do it with just hand watering, but an automatic mister is the best way that I found to and learned to um, root cuttings. Typically, you're putting it in a nursery setting with something like 30 to 50% shade. And then your irrigation timer, you want to use a propagation timer that has the ability to run a runtime like 5 to 8 seconds every 5 to 10 minutes. Typically, the way I learned was 8 seconds every 10 minutes for the first um, probably 2 weeks. And then as the roots start to generate, the shoots start to come up, you can kind of set it back to something like 3 to 5 seconds every 10 to 20 minutes. And then as the plant progresses, as far as rooting, you can cut back on that time. And then eventually you want to harden them off and then put them out in its own individual pots. So this image, I believe we were doing ulu cuttings here. Our nursery benches are built with these expanded metal on these old irrigation pipes, these galvanized irrigation pipes. We run PVC through the expanded metal with these mister nozzles, and they'll just come on. Uh, I think we were using a dig propagation timer for this one. It's a battery timer, and then you can set it to come on within a time frame too. And so it's not misting it at night when you don't typically need it. So we'll have a runtime something like 5:30 to 5:30. And then that'll come on every 10 minutes for 5 seconds or 8 seconds between that time period. Most regular irrigation controllers might have something like four runtimes. And so it can work, but it might not be as good. And then if you have a little bit more advanced irrigation timer, it can be something like four sets of four runtimes. And so you might have 16 runtimes. So it's like every half an hour you might be able to have a runtime. So at least something like that is better if you can't have or you don't have access to a propagation timer. Some things to remember, know your plant, that tissue type, the stage of growth it's in, whether it likes to be rooted as a herbaceous or semi-hardwood or hardwood material. Uniformity of cuttings is what I was taught. Make sure that they're all about the same length, about the same growth, about the same diameter. So when you put it out into the field, they're all about the same stage so that you have uniformity out in the field. And then maintaining a very healthy mother plant. The mother plant is the plant that you're taking all this vegetated plant material from. You want to make sure it's healthy and vigorous. So making sure it's getting adequate water, adequate fertility, and it's not full of diseases and pests. It's probably the best way to maintain your mother plants. And then this is just a cool image of some vegetative cuttings we were doing. for a grower that needed a specific variety of eggplant for their market, but the seed wasn't available. So we went into their field and we did cutting propagation of their eggplant. And so you'll be surprised what you can propagate. And it just takes a little bit of tinkering around to figure out the right ratios of media. So this, you can see some of that peat perlite left on the cuttings. The next one is air layering. So layering or layering in general. So basically we're rooting a plant with a part of that plant still attached to the parent or mother plant. Used typically for larger plant material. So if you want a larger tree with roots on it to plant into your field, layering is probably the way to go. Most times you're layering in the fall and spring when the trees are not in fruit or flower, or it's not in a vegetative growth and it's kind of dormant and ready to bud out too is another good time. So the types of layering include tip layering, simple layering, trench layering, mound layering, and then air layering. Air layering is probably the most common method, but there's other ways to do it. And so just wanted to put it out there so you have that exposure to it. So tips from the parent plant, you basically bury them in a shallow hole. New roots will form on that shoot. The shoot will pop out through the ground. Once those roots and the shoots are formed, you can then remove it from the mother plant and then pot it up or plant it out into the another area in the field. Simple layer is a little bit similar to the tip, but you're actually just running out a longer branch underneath the ground and letting that whole thing come up above the ground. Typically, you're wounding it about 12 to 18 inches below the tip. And so something like here would be your wounded area. You're anchoring this below the ground and top and filling it back in with soil in that hole. Your roots should generate from that cut area. And once it's rooted, you can then cut it completely off past the rooted area and then replant that. Trench layering is kind of the same thing, but you're getting more shoots off of that um, plant material because they're sending out a shoot from each individual bud. And you're typically doing this more with vining plants. But air layering is probably the most common form of layering or generating these roots on the plant while it's still attached. Usually you're using this for that really stiff hardwood material on some of those lower branches. When you're doing the air layering, you're making this 2 to 4 inch cut um, on that particular section that you want the roots to grow from. And so the best way to do it is to make 2 rings or completely around the branch. And then once you've made those 2 rings with a knife, you'll come back in with your knife and cut a slit across the 2 rings. And then you'll stick your knife in that cut and try and peel back the bark to make a clean cut around the bark area, exposing this cambial zone. And like we said earlier, that cambial zone has these cells that can regrow into itself or other things. And so one of the tricks is to take your knife and actually scrape this material to remove a lot of that cambial tissue and meristematic tissue from that area so you don't get reconnection of the vascular system from this cut end to that cut end. Once you've done that, you're typically going to get root production from this cut end uh, up onto here where you're putting your material. And so sphagnum moss is one of the main medias that I was taught to use. You're putting it in some kind of wrapping material. I like to use foil. You put the sphagnum moss in the foil, and then you wrap it around that cut end. And like I mentioned, from the top portion of that cut end, so this is the shoot part, you'll get a lot of root formation, callousing and root formation from this cut end, and it'll grow into that ball of sphagnum moss. Now that I've got access to pretty good quality core, I just been using coco core instead of that sphagnum moss, and it seems to work pretty well. So what I usually do is I'll take the foil, I'll wrap it around so I don't have to use ties around it. I'll just kind of make like a candy wrapper around the that cut stem end and kink the 2 ends. And then I'll actually come back in with saran wrap and I'll saran wrap the whole thing to kind of keep the moisture in. After the roots are formed, you can then cut the stem off somewhere down, probably around here. And then what I like to do is soak it in water for a couple hours to kind of generate the process of that water uptake by the plant. I also like to remove a lot of the top part of the tree because the root system is in this small ball. You kind of don't want to have more tissue above than that root system can handle. And so you want to kind of cut it back a little bit to kind of compensate for that uneven distribution of root to shoot. And then rooting hormone is another component to this process. And so I like to spread some of that rooting hormone on that cut end prior to putting on your foil wrapper with the peat moss or the sphagnum moss or the core. I like to sprinkle some of that on top of the core too before I wrap it around the cut end. In the Pacific, I've seen folks using just old recycled water bottles. And so you just cut a slit down the side of the water bottle, and then you wrap it around the base or the cut end of your tree. And then you fill this with, I've seen sand, I've seen ore, I've seen all kinds of things. And then you just secure the bottle with some kind of rope, tie, or string. And then in the Pacific, where I was, it was kind of rainy, and so all I did was let the rain trickle down from the branch into this to keep the moisture. And then once it's rooted in here, you can cut it off. That's another way to do it. I had some students come out to our research station a while back, and they were wanted to learn how to make air layers. So this is on citrus. Citrus is a pretty good tree to practice on because it roots pretty easily. And you can see here, this is all the leftover cocoa core, and all these little white lines are the root growth. And so each one of these are wrapped with a foil, and it's about two handfuls of core per foil ball. And then these are currently planted in the ground, producing fruit already, and it's about 3 years from this photo. The next one is division. Division is basically for plants that are only vegetatively propagated, like kalo, bananas. A lot of these tuberous root crops or have rhizomes are where we're using divisions. Basically, we're pulling it from the soil and then we're cutting several pieces from it and then forming these new plants that we'll put back out into the field. And then micropropagation um, or tissue culture. When I talked about that meristematic tissue, these buds, this is the apical meristem, which provides dominance over the whole plant. And this is that main growing shoot for that individual plant. A close-up version of that shows you this meristematic tissue. So a lot of these cells are still deciding what to be. And so it's a good place to create a new plant from. And so tissue culture uses this meristematic tissue to generate new plant material. And so some of the steps for tissue culture, assuming you have a really clean explant or mother plant, you can take a leaf from that plant, sterilize it, sterilize the media you're going to put it in, and then you take a small piece of that plant material, and you put it on uh, agar, which is like a gelatinous material. And then in some cases, that agar has other things in it, like hormones, like cytokinins, gibberellins, auxins. And then those are all from that one particular plant. You'll get generations of shoots on that material. And then you'll keep cutting those apart and putting them on there in new containers. And then once you've generated enough plant material, you can provide the auxins to it to generate roots. So these typically just have no roots. They're just sitting in this agar or gelatinous material. And then you provide it with the rooting hormone, and it's allowed to generate roots. And then from there, it'll look something like this. This is sweet potato tissue cultured plants in this gelatinous material. You can see the root formation. We open this up. We'll take out each plant material. We'll put it in something like a 4-inch pot and we'll allow it to harden off. So you want to make sure you're keeping that high humidity area for a little while till it's kind of acclimating to a lower humidity with a higher temperature. And then once it's hardened off, it can go into another pot or out into the field for planting. The benefits for tissue culturing is you can get a lot of plants really quickly. Once you've got this system set up, it does require a lot of specialized techniques and a facility to that's relatively sterile to be able to do this. So this is a laminar flow air hood, which you do a lot of this cutting in, so the air is pulled out from this area to reduce the amount of pathogens that can get into this chamber because it's kind of sitting in there. And so from one plant, you can get 1,000 plant materials from one leaf versus a cutting is one plant, an air layer is one plant. This allows you to generate thousands of plants from very limited amount of material. But it does require a lot of skill and a facility to do it in. And so it's not commonly practiced by individual operations. There are services out there that do tissue culture. Our lab in Hilo does offer some tissue culture services. I think Hark does some tissue culture services, and there's a bunch of companies, small companies coming up that are offering tissue culture services. Another benefit to tissue culturing is you can get you can maintain clean plant material in this system that you can distribute. If you're good, you can also remove some of the viruses from plants through this process because we're taking that meristematic tissue and it's so fresh that the virus from the plant might not have moved into that tissue. And so you can start with clean plant material going out into the field. And then macropropagation is another form of um, sort of division, but it's kind of utilizing micropropagation techniques, I guess, to generate a lot of plant material out in the field or in a nursery setting. So, banana macropropagation is probably one of the more common ones, I guess, or one of the only ones that I know of that is using or terming this phrase macropropagation. And so this is a chamber that I built for macropropagation. It's a wood structure lined with plastic, filled with cocoa coir. And what we're doing here is we're taking the corm part of the keiki and we're putting it into this humidity chamber that has restricted light. And we're allowing those tissues to generate more plant material than if you were to just take one keiki and put it out into the field. So the practice, what we're doing here is we're selecting bunchy top free plant material as much as possible. So each keiki that we're taking, we're doing BBTV screening on it using a test kit from Agdia. What we're doing here is we're boiling the corm to help remove nematodes, any fusarium or other fungal pathogens that are on the root end of the corm. And so we were boiling it for like a minute at a rolling boil, and the corm of the banana can handle that. Uh, Before boiling that, we would remove all of that dirt and debris to increase the amount of penetration of that boiling water into the corm portion. And then once we've boiled it, we'll come here on the table and we'll remove each sheath. At each sheath, there's going to be a bud. At that bud, we're going to basically kind of cross cut each bud down the center of that meristem. And if you can do it exactly down the center and split that meristem in quarters, you're potentially going to get four shoots from that particular bud rather than one shoot per bud. And then here, you can see the center of the corm dug out. And what that did was we've destroyed that apical dominance, so that apical meristem that's in the center doesn't have dominance over all the buds that are on the outside. And so that allows for all these other buds to break out and produce shoots. And so from that one corm, you'll start to see these shoots come out. And then from those shoots, we actually will, if you're confident enough, you can actually cross cut these again. And then this one shoot can potentially turn into four shoots. And so you can see here, this is one bud that turned into five shoots. And so you can cut these individual shoots into individual plants and then pot them up and send it out to the field. And so from one plant, you can get, we were getting maybe up to 15 to 20 shoots at the most per keiki in a couple month period. And so you can increase your plant materials for your field with very few amount of keiki versus trying to select keiki from the field from clean plants to plant your new field. This provides you with a clean stock to replant your new field, which helps you kind of run away from banana bunchy top. And that was a quick overview of different vegetative propagation techniques.